Hello everyone, I'm Tiago Pedreira. I am a PG student here at the University of Göttingen. I'm a PG student of Professor Dr. Jörg Stuttke. And today I'm here to, well, probably you don't know, but there is a new database for this synthetic microorganism, and I will give you a guided tour through the database. And so what is SynWiki? SynWiki is a relational database, and currently we have a collection of the functional annotation for this synthetic organism, the JCVI-syn3.A and also Syn3.0. Currently, we have over 400 protein coding genes. We also retain some of the gene names, some of the other ones we manually curated. But in general, we kept the same locus tag that you could already find previously. So you have the JCVI-syn3.A and then underscore and then the identifier of each gene. We've also created 66 functional categories, which are organized more or less in, well, we took inspiration from Go terms, but it's not exactly Go terms, but we wanted to keep it simple and short. Otherwise, we would end up with a lot of categories. We also started doing some prediction of protein structures using BLASTP. So we use the same protein sequence, and then we just blast against other organisms, and then we just annotate that. And we're also integrating some expression data. Currently, you can only find for one experiment from a previous study. And we've also added over 500 references across all genes. And again, all data is manually curated by us here in the lab. And you can find SynWiki using this URL. And in fact, our paper just got accepted a few weeks ago. And it's going to be, you can already see it and read it. And so if you use our data, please cite us. And now, better than just actually show you a bunch of slides with what SynWiki has to offer, we thought that it would be much more interesting for you to actually go with me on a guided tour. So I hope you can actually, can you see it? Can you see Mozilla Firefox now? All right, perfect. So if we just head over to SynWiki, this is the front page. SynWiki uses the same structure as SUKIWiki. For some of you who might know what I'm talking about, is for Bacillus subtilis. And so we try to preserve some of the functionalities.
Tiago, can you zoom in a little bit?
Yes, I can. Is it better?
That's better, yes.
Right, perfect. So some of the functionalities are what we call the browsers or the synapse, where you have genome expression, sometimes even interactions. And then we also have here a link for the labs. We have some latest news, also a citation that I just added. So let's start with the labs. We have just a collection of labs that we can obviously increase as more people start to work. You can just click on each person and then you can just send an email, go to the homepage, or even check the PubMed. And so it's obviously growing. All of this is currently growing. But let's move on to what I really want to show you, which is what is interesting, and is let's visit a gene page. So as I said before, we can either use the gene name or we can use the identifier, so the locus tag. I will just show you. If I just select this gene, for example, the PTSH, I can search directly using the gene name, or if we go back, and if I use the identifier, which is 0694, I go to the same one. You can use just the identifier. You can even use the whole locus. It's still gonna take you to the same page. So it's very flexible, and the idea is to make so that everyone who uses the same names or the same locus can always get to the same results. So what we have here, it's a table where you can find some generic aspects of the gene or protein. Here we have our genomic context, where it's usually focused on the gene that you searched, and then allows you to cycle through the whole genome. So you can click on other elements and go to that gene page. Here we have access to the functional categories that we created. I can show you roughly. So it's almost like a tree-like format, where you can just see through each category what are the genes that are under each one of them. We also added a list of homologs in different organisms, which we did using FASTA algorithm. And these are the organisms that we searched for. It gives you the protein name if it exists, the identity similarity scores, if it's bidirectional or not, And for those that we couldn't find, we just say that there's no significant homologs to be displayed. You have some generic aspects of the gene, about the protein here, as I said, the structure. We just say exactly how much identity there is. If there's a publication, we always refer to that. We have information on the expression regulation, which is, again, the data that we have integrated recently. And then we have a list of original publications or other reviews or other publications that we manually add to each gene. Here on the side, we have access to the interactions, for example, the structure, which is the same one. And all of this is highly interactive. So you can click on this one and you go directly to this gene page. I just wanted to show you, for example, here on the interaction, you can also click and you can move it around. In the future, we will also be able to integrate the expression and do an overlay on each view that you're trying to access. You also have here on the genome, which is a better view. It allows you to check for the DNA sequence, the protein sequence. You can query by flanking region. So I want P, T, S, H, and I want 1,000 up, 1,000 downstream. And then it will tell you exactly where is the gene that you queried, and then the upstream and downstream. You can also circle through the whole genome, or you can also just do from like actual coordinates of each gene. Lastly, what I want to show you is the expression, which this is still incomplete. We're still working on it. But here allows you to actually check for the data set that we currently have. It allows you to compare with other genes, like, for example, DNA, and then you can mouse over and you can actually see what are the protein levels on this condition, which is early growth phase. And then you can even download all the data. So you have here all the data and you can download. And yeah, so the database is very, it's still growing, obviously, and we count with the help of every one of you to keep generating more data. We also have here access to other databases that contain information for the protein or the gene that you're querying for. And obviously, for those that don't have, we just skip that. For example, this more unknown, this is actually a bad example, but if you have like RNA features, for example, those obviously will not have UniProt link, for example. And yeah, this is what I wanted to show you. And so we really hope that you continue contributing to this well, growing field, so that we can keep increasing the amount of data that we have in SynWiki. And if there's anything that you would like to see there, don't ever hesitate to contact us, because this is how we can grow our data in our database. And finally, I just wanted to thank everyone for listening and our SynWiki team. Thank you. Any questions? I can also stop sharing.
This has been fantastic, Tiago. I'm going to be sending you more data.
Thank you.
You know, hopefully you can link to this meeting webpage as well. So, and we will link to yours.
Thank you.
So, you know, we'll get all of this done, you know, very soon.
Yeah, thank you.
Others.
Very good. Amazing talk.
Thank you.
Important.
So in case you want to well, send us some data, you can always either contact me. I can put my email there in the chat, or maybe my boss, which you should have. I mean, yeah, on the website you provided, we have everyone's email address, right?
Mm-hmm.
Yeah, but I can still put it here. It's okay. So yeah, so just feel free to contact us to see how far we can, because all the data obviously has to follow certain rules so that we can integrate them. But it's definitely something that we can discuss and we can work on. So yeah, feel free to use it in the future. We really want to make this. So far, only people in the lab can actually edit the information that is there. We're very, we try to keep our eyes wide open so that we can fetch as much information as we can from latest publications. but if there's something that we're missing, please please try to contact us so that we can put that right away in the website. And in the future, the idea is actually to expand that for at least people that we know that have the knowledge, so that everyone contributes in one way or the other.
So I know the Paulson Lab at UCSD has done an in-depth transcriptome analysis of Syn3.A. You might be able to work with them to get that so you could assimilate this into your expression data.
Yeah. So the idea is obviously this is a project like this is something that it's always growing, so we can't just put everything at the same time in the database. So this is something that obviously we're working every day. We try to include as much data as we can, and obviously every time that we see a new study, we always try to address that data and put it as fast as possible in our website.
Thank you very much. For our whole community, this is a fantastic resource that you have given us.
Thank you.